Hi everyone, this lesson is going to be focusing on vaccines and vaccinations, very topical at the moment, so I thought we would start off with looking at this infographic from UNICEF, and it's really quite powerful. It talks about what can be prevented from using vaccines. The main thing is death, and the other thing you can see here in this image is a blue skin. So this is a really great article to read about why we learn about these things and why it's so important. This man was self-medicating with a substance known as colloidal silver, and it caused his skin to turn blue, and sadly he eventually died. So learning about this stuff is really important. All right, so these are the key words that you need to know for this lesson. Quite a lot going on in here. It is very topical. I would expect this certainly to come up in your exam. Words that we need to know are vaccine, attenuated, toxoid, subunit, recombinant DNA, which we will visit again in the next topic, immunization, natural, active and passive immunity, virus and virulence. So there's lots of keywords here, and you definitely need to get a handle on them. Learning objectives seem quite short, but this is quite an involved lesson. Defining what a vaccine is and describing the reasons for vaccination. Hopefully in today's climate you'll be very aware of that. And looking at the 4 types of vaccines. So if you've been keeping up with the news and the podcasts that I've been directing you to, they have been talking about vaccines quite a lot. There was quite a lot of slides here, and I decided just to condense it down to one page for you. And this is showing what can happen if you don't get vaccinations for very preventable diseases. So we have tetanus, rubella, measles, mumps, polio, and HPV or genital warts, which can affect both males and females. In females, we have the vaccination for that in schools, and that is to prevent cervical cancer. This is a short and not an exhaustive list of things that can apparently cure HPV. The second last one is my favorite, and no, they don't work. Okay, so how did vaccinations come about? Well, this guy here, Edward Jenner, he discovered vaccinations for smallpox. How he did that was a really interesting story. So if you are logged into Twig, please pause this video and click on the link to watch how Jenner started the eradication of smallpox as we know it. Okay, welcome back. Hopefully you saw that video, and I found it pretty inspiring. I think it's amazing that you have pioneering people like that that can actually go out there and use their inquiring minds to save millions. All right, so looking at the syllabus points here, there are 2 types of immunity that you can get or acquire. You can have active immunity, so as that name suggests, your body is actively making its own antibodies and memory cells. Or we can have passive immunity. So the word pass at the front, just if you imagine someone's passing you ready-made antibodies. If we look at this, actively acquired immunity can be natural. So you become infected with a disease, you thankfully recover from that, you make memory cells from your B and T cells, and they will stay in your body for a number of years, or even up to your lifetime. The way that vaccines work is by inducing artificial active immunity. So we inject people with a non-virulent version of a pathogen. So when I say non-virulent, it doesn't cause the disease, but it still stimulates that immune response. So crucially, it makes memory cells for us without actually having to suffer the disease in itself. So this side of our branching key, we get memory cells, and that's a very important distinction here. On the other side, we have passive immunity, and that can be natural. So this is when there is no human intervention, there's no doctor putting a shot in your arm of ready-made antibodies. We get these things across the placenta and also through breast milk. So that's why breastfeeding is considered to be very important. It supplies the newborn baby with antibodies because it doesn't have a fully functioning immune system yet. So I want you to be thinking, the type of exam question you would get would be to compare these types of immunity and perhaps to discuss which one is more effective and to give reasons for why. All right, so again, this infographic is just looking at different types of immunity. I'd like you to look at each of these images and to decide if it is natural or active. Is it passive or artificial, or is it a combination of the two? All right, if you've had to think about it, let's start off with this image of a needle. We're imagining that this has got some sort of vaccination in there. So that, of course, is not a natural method. That's an artificial method of vaccination. And it is active if it's an attenuated or destroyed virus that's stimulating your body to produce its own antibodies. If it is a serum with antibodies in it, it would be artificial again because it's got human intervention involved, but we're simply passing antibodies to someone. So that would be artificial passive or artificial active. Definitely artificial, not natural at all. These other two images, we can see that they are both in the natural camp. The antibodies that the baby receives from breast milk, we would say that is natural and it's passive. This poor wee lad here is suffering from what looks like some sort of pox, chicken pox. That is natural type of immunity because he survives, and is active because the immune system is stimulated to produce its own memory cells. This table is just a summary of what we just talked about, something that you should definitely know and commit to memory. Make sure that you understand the difference between active and passive, natural and artificial, and how each of these individual 4 boxes are distinctive from each other. All right, at this point, you should be able to define those 4 different types of immunity. So pause the video here. I'd like you to complete page 38 of your class booklet. If you haven't already done so, you should definitely read page 151 of your textbook to help you achieve that learning objective and fill out that page in the booklet successfully. Okay, welcome back. We are going to be looking at the differences between immunization and vaccination. These words do tend to be used interchangeably, however, they are different. If you want a bit of a background before we talk about this, pause the video and click on that link and look at the short twig video on vaccinations. If not, you can look at it later. So, an immunization. This is when you become immune or resistant to a particular antigen. So we're looking at foreign antigens here. This can happen naturally when you are exposed to a disease, or it can happen artificially when you are given an injection. That antigen is introduced artificially via some sort of medicine into your body. A vaccination, on the other hand, is when you are given the antigen via a vaccine. So this is via a medicine, and that makes your body become immune. You build immunity without having to have contracted the disease itself. So that's what the key difference between immunization and vaccination is. You can become immune to a disease by coming into contact with the actual live disease. With a vaccination, you don't come into contact with the live disease, but you develop immunity anyway. Okay, so this is just a little infographic to summarize what we're talking about in the difference between vaccination and immunization. Vaccination refers to the process of giving a vaccine, so the little syringe icon should help you with that. The vaccination is introducing the foreign pathogen that has been attenuated, but it has the antigens on the outside. And that will stimulate your body to react. And it will go through that process of making antibodies. So think about what type of cell that is. Is that a B or a T cell? Hopefully you remember that is a B cell, which will then produce plasma cells, which produce the correct antibodies. So that process will lead to immunological memory, remembering that these plasma cells will clone and produce antibodies, whereas some B cells remain as memory cells, giving us immunological memory to that specific antigen. So if we ever come into contact with it again, we will react fast. Now, immunization refers to the process where the immune system recognizes that foreign antigen or protein, and we produce antibodies and other immune reactions against it. All right, so just to give you some terms that you can write into your booklet. So this is page 42 of your booklet. I sourced this information from pages 152 to 155. These are the key terms that you should know. Please pause the video, write in those key terms, and read those pages in your textbook. All right, thanks for reading those pages, guys. We are now looking at something called the primary and secondary immune response. So I'm going to talk you through that. This is something that I would really like you to be able to write maybe a 4 or 5 mark answer to. Now, if we look at the start here, at time 0, there is no line for the graph. We haven't been exposed to any pathogen, therefore we have no antibodies. At this point, so let's say this is day 5, we start to see an antigen-antibody reaction take place. So what actually happened was at this point where the arrow is, there was the initial exposure to an antigen. Now the reason why we don't have antibodies being created straight away is because we have to go through that process of clonal expansion, production of the right antibodies with the correct receptor sites that will fit with the antigen. Now, what you should be noticing about this response, this primary or first immune response, is that it's quite short-acting and the concentration of antibodies is actually really low. And that means the first time we are exposed to a particular pathogen, we tend to get quite sick. We tend not to fight it off as well as we would hope. Now, over time, once you recover and get better, you'll notice that your antibody levels in your blood serum, they never go back to 0. So thinking about that, why do you think that is? What has been created here? You should be aware that you have created memory cells, and these will stay in your blood. So if you ever become exposed to that antigen again, which we have at day 28, you can see that the immune response occurs much faster. So you have this rapid rise in the concentration of antibodies in the blood. So it's a very rapid reaction. It's very long-lasting as well. So you can see that the concentration of antibodies remains very high for an extended period of time. So if you do ever get sick again from the same pathogen, you tend not to feel the same symptoms. You tend to actually feel pretty okay, and that is because of this secondary immune response. Now, this here is referring to how the body will react to a dose of a different pathogen. So I want you to be thinking about why we go back to the primary response. Why is that different to this blue line? You should have recognized that because we have different pathogens, we have to make different antibodies. So every single time you meet a different virus or bacterial toxin, what actually happens is your body has to create antibodies that specifically match in a lock and key mechanism with that pathogen antigen on the surface. So unfortunately, we have to start the primary response again before we become immune. All right, so just to summarize, the primary response takes time, it's a slow build-up, and it's not very long-lasting. The secondary response, you can see in this graph, classic graph here, and these little cartoon diagrams, because we have memory cells, we can mount an immune response much faster because we have a large number of antibodies that can be produced quickly, and they will destroy the pathogen, and therefore we may not even experience any symptoms. Okay, so this is what we looked at the start of the lesson. We are going to be focusing for this part of the lesson on active immunity. And we are going to be looking specifically at active artificial immunity. So again, very topical, we're looking at how we can develop vaccinations to fight against certain diseases. Now, the reason why it's important that we learn about the immune response is because B cells, specifically B cells, are key to the primary response. Because they produce plasma cells, they therefore secrete free antibodies that will inactivate the pathogen. So remember, there's 4 ways of inactivation, but it's only plasma cells that secrete free antibodies that will do that. Okay, so this checkpoint refers to the learning objective that asks you to compare the primary and secondary immune response in attacking an antigen. Please stop the video here and complete page 31 of your class booklet. If you haven't already done so, please do the pre-reading, page 148, to help you understand and write a full answer for this learning objective. Okay, hi guys. We're going to be looking at 4 different types of vaccines that you should know about. So in broad general categories, this is what we're looking at. So you have live attenuated vaccines. So the word attenuated means that the virus has been destroyed in some way. If you look at this cartoon of it here, we've got this virus particle. So we know that it has some sort of nucleic acid. Here it looks like RNA. I know that because it's single-stranded. It's wrapped up in this protein or lipid coat, and it has these antigenic markers on the surface. Now, an attenuated vaccine will have these antigens still intact on the surface. However, there'll have been some sort of damage to the nucleic acid, which means it cannot reproduce and therefore cannot do the body harm. Common ones that we use at the moment are TB, that's the BCG injection that you may have had, polio, measles, and other ones that you may or may not have heard of, rotavirus and yellow fever. Now, that is a live attenuated vaccine. We also have an inactivated vaccine. So this is when the entire antigen has been killed, and that is to induce, again, the active immune response, but again, keeping the human body safe. The third one is the subunit or purified antigen vaccine. So pneumococcal, hepatitis are the more common ones. If you've ever been to countries in Asia, you may have had one of those vaccines. So a subunit vaccine, that is where we manipulate the DNA. We will talk about that in the biotechnology topic in semester 2, but essentially what we may do is take out some of the viral DNA, and we will amplify that and use that to stimulate an immune response. We also have toxoid vaccines. So a toxoid refers to an inactivated toxin. So bacteria secrete toxins. If we produce a toxoid that is mimics the toxin, but it is unable to make you sick, we can then mount an immune response to that. So just to summarize, there's two broad categories that you need to know about. There's whole agent vaccines. So these are vaccines that contain a safe version of that microorganism. So that means it's not going to make you sick, but it will cause that immune reaction. These whole agent vaccines can be inactivated, which means that they are genetically switched off, or they can be modified live viruses, and they are attenuated, so again, they cannot harm you. You then have subunit vaccines, and there's three types of those. There's the toxoid ones, which refer to pathogens caused by bacterium, acellular vaccines, and genetically engineered ones. And the genetically engineered ones are certainly where we are going in the future, and certainly with COVID-19. Okay, so whole agent vaccines. What we are looking at here is the infectious agent is involved in the vaccine. It's usually treated by chemicals so that it's being killed. When we talk about it being chemically treated, we normally say attenuated, and that means it can't harm you anymore. And that's commonly what you'll be getting with your flu shot. We then have the modified live virus. It can no longer produce a clinical disease. The virus in these vaccines can replicate within the host, and that will give a really great immune response because your body will actually think it's under attack. But you can see in this cartoon here that it has been modified to not cause any sort of problems to your body. All right, we're looking at subunit vaccines here now. We have the acellular one. Now that is when an immune response is stimulated to develop antibodies against a whole virus, but because no living virus is actually present, subunit vaccines are considered to be safe. So we are not introducing a full virus into the body and just rendering it harmless. It's only part of that virus. Toxoids, talked about them before. These are bacterial toxins. So these are the chemicals that the bacterium releases, not the bacteria itself, so do not say that in an exam question. These have been inactivated. So these toxic chemicals are inactivated by chemical or heat treatment, so hence why we changed the name to toxoid, and they help your body to raise antibodies against things like tetanus and diphtheria. Finally, genetically engineered vaccines. These are when part of that genetic code from the virus is introduced into a simple organism like a yeast or a bacterium. It is reprogrammed so that it will make the antigens. So you can see here, this, let's say it's a simple yeast cell. It has part of that viral DNA inside of it. And it's just the part that makes the antigens. So these antigens will appear on the surface of that harmless yeast or bacterium, and that will stimulate an immune response. All right, so looking at why we need to have booster shots. So once you've had a vaccine, why do you need to keep getting more? And there's some vaccines where you'll need to get up to 3 shots to ensure that you are completely safe. The reason for this is that we want to ensure that you have a high enough antibody and memory cell level within your blood plasma. The vaccination is provided to ensure that there is a decreasing number of cases of the disease. So we want to try and eradicate a disease, and that goes back to smallpox. The immune response is normally sufficient to give us memory cells. However, you can see that sometimes the concentration of antibodies in our blood serum will dip. So by having that booster shot, it will trigger memory cells to raise our antibody levels to a higher level and keep us nice and safe. So if we ever do meet that pathogen, we have a high concentration of antibodies ready to go. This graphic was from a magazine called Nature. It's a really coveted or really famous science magazine. This is current research that's out just now about COVID-19. And I put this in here just to show you the different types of vaccinations that the scientists all over the globe are looking at. You can see here that only a small percentage are looking at live attenuated vaccines. But a lot of the work is actually going on to more complicated biotechnology. So we're looking at DNA, RNA, peptide-based recombinant proteins and vector-based vaccines. This is all to do with manipulating the nucleic acid. Now that we have an understanding of the genomic sequence of COVID-19 and other viruses, we can then manipulate that, hopefully in the future, to come up with vaccines that could lead to immunizations. From that article, I just pulled out some information that I felt was really pertinent to what we're learning about. The vaccine development for COVID-19 is using a range of technology platforms. So it's including nucleic acid and virus-like particle peptide and viral vector recombinant protein technology. This might seem overwhelming with a number of different words in here, but as we go through the course and look at the biotechnology topic, hopefully you'll understand the power of this research and how it could potentially save millions of lives. Okay, guys, again, another checkpoint. So we had to describe 4 types of vaccines. I gave you a 5th one for the subunit ones, so you can just pick one from there. Please pause the video, complete pages 42 and 43. And again, if you haven't read those pages in your textbook, please go back and read, or better yet, reread pages 152 to 155. All right, so that was the end of the first lesson. That's what you need to know about vaccines. What we're going on to now is looking at the reasons for vaccination. These videos were done by the National Health Service in the United Kingdom. These look at explaining to the general population why vaccines are important and why we get them. Now, you know that there's a lot of misinformation out there where people feel that vaccines aren't necessary and it's some sort of a government ploy to control the masses. What I would say to that is it's important that you have a very thorough understanding of what vaccines are, how they work, and the potential they have to save lives. It's something that could possibly come up in your end of year examination as an extended answer because it does touch on ethics. So please watch these videos. Once you've done that, there are a series of other links to watch. Now, I am going to stop the lesson here. What I would like you to do is to work through this content yourself, and by engaging with your peers in the class, and you can even have a discussion with me about this, to look at herd immunity, why vaccinations are important, and how they have affected world populations. So here is some information that I have put in for you to get you thinking about it, and then you can look at what vaccinations do, and there's a lot of different scenarios for how you would respond. So I want you to be thinking about this from both sides of the coin. Okay, guys, that's the end of the lesson. If you have any questions, please email me or log into Teams.